really excited for today, and I would like to kick off our event by welcoming some very special guests, and in particular Susan Ferris, Virginia's daughter, and her husband Terry. You are sitting in some very special seats. And we have all agreed that you're allowed to ask the first question. No expectation. Interim Dean Foster sends his regrets. He would have liked to have been here, but unfortunately he cannot. I'd like to welcome our guest speaker, Dr. Hongmei Libiali, just yet, and to welcome our award recipient, Hannah Turner, who's sitting back there.
Can we applaud?
I'd also like to welcome April Shepherd from Development. I'm not quite sure. Hi. Oh, there she is. It's great to welcome you to the department. Yes. Thank you for the bug today. Well done. We are going to do more of that. And then I'd also like to welcome back Mardell, who has been absent for a while. Please don't do that again.
I got one more hip. So.
All right, so today is a fun day and it's a very special day. I think that we have been waiting to celebrate this day for several years. We gathered really to celebrate the legacy of Dr. Virginia Ferris, who was a valued member of the Department of Entomology and the college. She joined us as a faculty member in 1965. She was an esteemed scientist, a highly regarded nematologist, and she was most definitely a pioneer and a trailblazer. She was also an administrator. She had some funny comments for me about administrators, which I won't share with you today. Something about the light going out. And a valued academician colleague, and most importantly, a mentor. And I'll come back to that in just a moment. We're also here to recognise some early career scientists, some emerging leaders, future leaders in their fields, both in Dr. Hongmei Libiali and in Hannah Turner. I will say that Hannah, like Virginia, was a graduate of Wellesley College, and Virginia earned her PhD from Cornell, and we've discovered at lunch today many interesting connections to Cornell. In a few moments I'm going to pass the floor to Dr. Matt Ginsel, who's the chair of our Department Research Committee, and it was the Department Research Committee that identified and nominated Hongmei as our guest speaker, and he will say a few words and remarks on Virginia's career and her accomplishments and contributions. But I just want to say a few words because in preparing my remarks, it became apparent to me just how incredibly accomplished she was and what she really gave, and I hadn't realised until I saw it really in front of me on paper. She was the first woman, a faculty member appointed to the College of Agriculture. She was also the first woman to purchase a ticket to the Ag Alumni Fish Fry. She was also one of the first to apply modern molecular techniques to the study of nematodes in her field, and so she subsequently became a pioneer in that area as well. She developed and ran a successful consulting business, not on campus. She pioneered the Cystex technology, so it's based on resistance traits to preventing damage to crops from the serious nematode pest. She really achieved so many wonderful things throughout her long and successful career. She was recognised as a Fellow of the National Science Foundation, a Fellow of the Society of Nematologists, a Fellow of the European Society of Nematologists. She is also a recipient of the Distinguished Ag Alumni Award from the College Ag Alumni Association. But I think, and what became really apparent as we were meeting and sharing so many amazing stories of Virginia at lunch today, was her gift to young scientists and her investment in the future. And that really has come through in so many different ways, because so many of us here, whether we are students or faculty, received mentorship and guidance and support from Virginia. So she contributed to so much. And we see that, we're so grateful to help that legacy continue through this award. So we're very grateful to Virginia and her family for this board is that it enables us to continue her legacy of investing in future scientists. And that's a really amazing thing. So that's why we're so excited. So I'd like to briefly hand the floor to Matt, and then I will introduce others.
Thanks.
Are you going to talk about basketball tickets, Matt?
I won't. I could for a very long time. Virginia and I shared a mutual passion for Purdue basketball. But, I prepared, yes, spoiler. I prepared a few remarks, and it's my pleasure to reflect with you on Virginia's career, accomplishments, and scientific legacy. In her more than 50 years in the Department of Entomology, she was a renowned scholar, the world's leading expert on soybean cyst nematode, and was an indelible influence on the research, teaching, and extension missions of the university, and on the culture of our department. Virginia's passion for science began early in life. After high school, she packed her bags and consuming interest in biology and left Abilene, Kansas for Wellesley, Massachusetts, where she attended Wellesley College. And so at this all-women's college, she found a competitive environment, academic environment, female professors that served as role models, and the self-confidence to more than hold her own when she entered Cornell as a graduate student in plant ecology. After earning a PhD, she joined the Cornell faculty as an assistant professor and began her research in nematology. It was there that she met and married John Ferris, who was a major collaborator of hers throughout her life, and throughout his life, and her best friend until his death in 2000. And so the couple left New York in the mid-50s and headed to the Midwest. And it was with Virginia's last paycheck from Cornell University that she bought a microscope, and she used it. It was difficult for women to attain faculty positions, especially in this region at that time. And so she set up a lab in her home and went to figuring out what's going on with these nematodes and did research as a freelance consultant, first in Illinois and then later in West Lafayette. And then John joined the faculty in 1958. And so with their two children in tow, and I'm sure Susan can tell us more of these stories, the Ferris traveled extensively, collecting in different species of nematodes throughout the region, often camping. Virginia was a wealth of knowledge when it came to Indiana campgrounds. And she gave me great advice. And what I found is the campgrounds that I love, Virginia also loved. And at times even processing some soil samples in hotel bathrooms. She was already one of the foremost experts in nematology when she was appointed to the entomology department in 1965. And as Kate mentioned, she was the first female faculty member in the College of Agriculture. She became a full professor 9 years later, and along the way served as assistant dean of the graduate school, assistant provost. She served as president of various societies, including of the Society of Nematologists, and was associate editor of the Journal of Nematology, recipient of the Helen B. Shulman Gold Medal Award, which honors individuals for their meaningful contributions to the promotion and advancement of female students. And that's really one of the hallmarks of Virginia's career, was her mentorship. She was a Phi Beta Kappa, a fellow of the National Science Foundation, the Indiana Academy of Sciences, the Society of Nematologists, and the European Society of Nematology, and was awarded the Purdue Agricultural Alumni Association Certificate of Distinction in 2017. Few careers show the breadth and reach and stretch of scholarship at Virginia. And as I mentioned, mentoring was a hallmark. Throughout, she was a tireless mentor and a champion of budding professionals, scientists, and young faculty alike. She encouraged generations, particularly women, to intellectually push themselves and to pursue their passions with intensity. On a personal note, I gained great strength from Virginia's wisdom. I count myself lucky to have known her and to have her rooting for me. She was a colleague, a mentor, and a friend. And like all that knew her, I miss her greatly, but I'm encouraged that her legacy continues. It continues through those that she touched, and it continues through this memorial endowment.
Thank you, Matt. So we've now now like to introduce our guest speaker, Dr. Hongmei Lee Bialy from Central State University. Many of you know Hongmei, and you know that she's an alumnus of the department. She received her PhD working with the program of Dr. Gary Pittendry, working with Drosophila. And then from there, you went to the University of Illinois Urbana-Champaign and joined the program of Dr. Jean Robinson, and thus began your work in honeybees and bee genetics and genomics, which continues to this day. Subsequently, you went to NC State, is that correct? Yes, and continued the work there. And then started your own program at Central State University, and conducting some amazing research, and also a highly valued instructor, teacher, and mentor yourself. I think you also interacted many times with Virginia, and I suspect she probably mentored you just as she mentored so many of us in this room. So, Hongmei is giving a talk entitled Celebrating Women in Entomology. I'm dying to hear what you're going to talk about, Hongmei. So, without any further ado, thank you the floor to help us celebrate. I'm going to try and place this again.
Thank you so much, Kate. I'll scroll my slides first. Yeah. Well, thank you so much. I am so grateful to be invited today as a guest speaker. So last time I was standing here and gave a talk was back in 2007, and about 15 years ago. So right now I feel like I'm riding a time machine. Oh, back. Feels so good to be back home to our department. So yeah, I'd like to start my talk with the opening to share my memories of Dr. Virginia Ferris. So I came to Purdue back in 2002. It was August 16th, 2002, on a rainy day. Got off Purdue Airport and started my PhD. I was very excited, and at the same time very nervous. And one of the first classes I took is actually Dr. Ferris' class, molecular systematics. And before I started at Purdue, I didn't really have much experience in molecular techniques and molecular biology. So her class was definitely the first one, opened my eyes and teach me everything about molecular biology, genetics, evolution. So my memory with her, I want to share, it's about her passion for science. Actually, Kate just mentioned earlier, every Thursday, our department had seminar talks. We have speakers and give a talk. And by the end of the talk, Dr. Ferris will always sit in the front row, and she would always raise her hand as the first one and ask really compelling, intriguing questions. And when I look back now, when I was a graduate student, I didn't really think that much and just think, wow, she has a lot of questions. But now I look back and I realize, oh my God, what an amazing, colleague and scientist. Because the best thing you can give to the researcher, to the speaker when they come to give a talk, it's your thinking, it's your engaging, it's your thoughts on the topics, really compelling discussion to make them better. And she did it 1 year, 2 years, she worked here more than 50 years. I've never seen any woman has such a passion for science and for research like her. So every time, I go to seminar talks now, I always think about Dr. Virginia Ferris. And I want to be like her as well and do a deep thinking of the talk and give my thoughts to my colleagues. And I also take her class, and we also always talk in the hallway. And she was always the most amazing and energetic woman I ever know. And one time we were talking about scheduling and time management, and she just mentioned quietly or pretty calmly, yeah, I go to sleep at 7 p.m. and I get up at 3 a.m. and start working. And I was a graduate student, you know how graduate student life goes, right? Most people stay up, I don't know, 1, 2 a.m. and I'm like, oh wow, when I went to sleep, you got up and started. But it was so amazing, you know, to have her mentor from the classes, from my research. So by today, still every day, I use molecular techniques for my research. So the next I would like to okay, before I share my research with you, I also have a couple pictures. So these two are actually Virginia Ferris graduate students. In the middle, this is Shujuan Lee. So she read Airplane with me together when we came to Purdue. So we were the same cohort, and she's definitely like my sister. And this is Virginia's other graduate student. Her name is Lu Sun. She's our like senior graduate student in the department. So, but both of them right now, Shu Juan is a really outstanding extension specialist at University of Arizona, work on pest management with the community, and Lu is an outstanding scientist working at National Center for Biotechnology, Information, and CBI, working on genomics. So this is another picture. I told where's Dr. Steve Yannick? So I told you I'm going to have a picture of you. And this is, everybody know this where is that, right? It's like the unit, right? So, and you know, recognized without stage. And so Shu Juan won the Teaching Assistant Award in that year. And so she kind of like provided me all the pictures. To show Dr. Virginia Ferris sitting there, Dr. Chris Aceto, and Dr. Steve Yannick sitting there. Very nice picture. So yeah, so with this, you know, think about the knowledge she taught me, the scientific research she mentored me. I'd like to share my research with you today. So in my, throughout my career, I focused on the honeybee genetics, epigenetics, behavior, physiology in general. And today's talk, I'd like to just touch base on three of the themes here. Normally people can only remember 3 things, so we're trying to not jam into too many things here. So I'll talk about some DNA methylation work I've done. And the second part is on the behavior and brain metabolism. And the third part is how I initiated research at Central State University as a new land-grant research. So what is DNA methylation? The DNA is the genetic material in every organism, also for bees. So the cytosine is one of the 4 kind of bases in DNA. On the position 5, if there's a modification of CH3 methyl group added in there, we call this a methylated cytosine, right? So it can be a part of 5MC. And so on these marks on the DNA are actually very important because these marks can be a signal to tell the cells and about give downstream signals on the cellular activities and gene regulation. There are also important enzymes that are associated with DNA methylation in the honeybees as well in like all the insects here. So this is in one of my review papers in the honeybees, Apis mellifera. The most important are DMT1, so DNA methyltransferase 1, and then DNMT3. So one of the highlight research I want to mention is that when I was doing postdoc work at North Carolina State, together with my collaborators, we were able to infect the honeybees with the viruses. It's called an Israeli acute paralysis virus. But with this infection, I was curious to see how DNA methylation can relate related to honeybee infections. And we treated the bees with viruses after 5 hours or 20 hours or 48 hours. The PBS is like a sham control. The IAPV is a viral infection there. So it's interesting, after 5 hours, it's kind of beginning of the infection. These are the expression levels of the enzyme DNMT3. However, after 20 hours, the virus start to take off, have a negative impact on the bee. These are pupa samples. We see a dramatic drop on the both DMT1 and the DMT3. And then by the 48 hours, we see really dramatic drop as well. So really interesting thing here in addition to the enzymes is that we see the methylation changes on a lot of the immune response genes. So a lot of these genes are involved in different immune pathways. One of the interesting ones is in the APK signaling pathway. The gene is GB43638. We see a dramatic, really decrease in their methylation at the beginning of the infection. And then we see actually a increase by the 48 hours. So, but the gene expression level really decreased dramatically by the 48 hours, right? So I think there's an indication here that methylation maybe take off first as a signal in the cell, and then throughout the time, transiently, by 48 hours, it will finally work on the gene expression level. So another topic on the gene methylation I was interested in is in gene regulation. A really important mechanism for gene regulation is called alternative splicing. So this is a human example, but same in the honeybee's genome. We can have different exons in the gene or different introns. So after the splicing, the introns most time get purged out. You have exons 1 to 3 kind of merged together, and that's a matured messenger RNA. And then that can be translated into protein isoforms. There are different ways for splicing. So you could have exon 1, 2, 3 together, but you could also have only exon 1 and 3 to skip exon 2. And because this variation, because splicing, we could have way more protein isoforms in the genome in this way. So the research question on this part is really look at the relationship between the DNA methylation and the alternate splicing. Will the DNA methylation affect the alternate splicing? If we knock down the key enzyme, DMT3, what will happen? So we want to test the hypothesis here. We think the methylation can impact the gene regulation through where the alternate splicing. So this paper from my postdoc work, we did a knockdown of DMT3 in the honeybee samples. Overall, every single trial showed a knockdown of the DMT3. And then we look at the splicing patterns. So this is one of the examples here. When you have the exons in these long rectangle, the lines are intron. So the red one is the exon, like exon 2 I mentioned earlier, right? This can be capped or can be skipped. And we also look at their methylation. So when in the knockdown bees, the methylation level really decreased, like 4% around this area. And we can see that what happened is the significant difference on their exon skipping event in this case. So I think, and just really quick summarize, we see a decrease of DMT3, the enzyme that will decrease the methylation, then also affect the alternate splicing. And besides exon skipping, there are also other events I just don't, probably won't have time to mention here. But the really cool thing is that we were able to knock down the enzyme to manipulate the methylation and then look at the changes in alternate splicing. The ultimate goal is to study how this affects the phenotypic plasticity in the honeybees or social insects. So switch gear to the second theme I was talking about. I have some research on the behavior and brain metabolism. One of the interesting behavior, so honeybees are social insects. They have 50,000 bees live in a colony, and if you can imagine you live with 50,000 people at home, at one home, would that be crazy? Right? You probably can't even get a one night good sleep. But the bees can do it. How do they do it? They have a whole repertoire of social behavior to organize a society, to make decisions. So aggression is one way to look at their defensive behavior. How do they defend a colony? Or how do they respond to intruders? On the right here, this is used to be called Africanized honeybees, but I just want to call them aggressive honeybees. They're Apis mellifera squalata. And these aggressive honeybees are called killer bees also. They're escaped from Brazil and always want to try to make their way north. These are the European honeybees here we see, which are Italian bees. So for aggression, it's really interesting. The killer bees can be really aggressive. If you kick a hive, thousands of them gonna come out. You run for a mile, they're still chasing you. The European bee is not much. So we wanted to ask the question, what to make these bees so aggressive? Is there anything to do with the brain metabolism? So I have these 3D printed bee brain to show everyone. So honeybee brain is, I think it's complicated, and they have different parts in the brain. Especially like the top part blue here is the mushroom body for learning memory. So for aggressive bees, we wanted to know in the brain of the bees, is there anything about their metabolism in the OX-phos pathway? So that's oxidative phosphorylation pathway. It's in the mitochondria, it produces ATP for the brain. So we want to ask the question, if we block some part of this pathway, would that make the bees more aggressive? So I'm going to show you a really quick video to see the assay. We drop an intruder bee, a girl worker in the cage, and we just observe to see how the existing bees respond. So this intruder is from a different colony, so she may smell different, but you can see that other worker in the existing cage grab her, try to attack her, use the antenna. So all these behavior listed here can be measured when we observe these colonies, and we can give a score of the colony, whether they're highly aggressive or not. And we use the two inhibitors here. So that's fenparoxamate and the tetraedifen. So this target the complex 1 in the OX-phos pathway, and this target complex 5. And the results is showing when we have the drug-treated one, they have increased aggression compared to the acetone control treated bees. So we wanted to have transgenic bees, but we don't have transgenic bees. So we're hijacking the fruit fly system. That's also the system I use for my PhD. So we hijack a fruit fly Drosophila system. Basically, we can manipulate the Drosophila genes and knock down the genes we're interested in the pathway, in the OX-phos pathway, and observe the aggression in the flies. Of course, the flies cannot sting, but they have this lunging behavior, which is really interesting. So you can see these are male. This is a winner fly. This is a loser fly. The winner fly can stand on the back legs, and really hammer down attack the loser fly. And we have, luckily, we have an automated system to automate the behavior. So purposely, because the transgenic fly system, we can make the knockdown of the genes expressed in the brain, especially in the neuron cells, by special drivers. So the neurons are these, these are two different neuron pictures here. And it's like ride a train, from here, Lafayette, West Lafayette to Chicago. And you have signals in transmitters transmitted within that way. And so we want to study whether when we express these genes and manipulate their gene expression, would that have anything to do with aggression? So these transgenic flies, when they have the LV driver and their RNA donor, and these flies they have decreased gene expression of ND22. That's NADH dehydrogenase, and they have increased number of lung genes compared to their parents. And in addition, we also want to test other tissues besides neurons. So we test the glia cells. The glia cells are these supporting tissues in the brain. And so it's like, think about the train station I talk about from West Lafayette to Chicago. You have train tracks and you have different railways. So the glia cells are like those railways, kind of separate all those signals. But we didn't find a really significant difference in these comparison when we look at the glia cells. So in conclusion, OXPHOS pathway seems to regulate aggressive brain, and we kind of know the neuron cells work in that way by manipulating in the fly system, and there's a really different, distinct brain state. The other behavior I want to talk about is the grooming. It's also mite biting behavior. So Brock Harper here studied mite biting, also with Crispin Gibbon, doing the breeding system for so many years. The video here is showing you how the worker bee come up. They really grab the mite. There's a live mite on the tweezer. And so the thought behind it is that the mites will have a damage and drop to the bottom board. And then we can collect the mice from bottom boards. So my students, Jada and Saren, helped collect thousands and thousands of mice. And we look at them under the microscopes. And so panel B is what we're looking at, we're looking for. We want to see how many of these mice have missing legs as a damaged mite. So it's an extended phenotype to measure their mite biting behavior. So the commercial bees are from Ohio. The mite biters are from Purdue here, actually. The mite biter open mated are the virgin queens mated in Ohio with the feral colonies. Supposed to have a high biting also population. So we see significant difference here. Both the Purdue breeding queens and the open mated ones have a high mite biting behavior. So in addition, we want to dig into more on the mechanism. So how do the bees do this? They actually use the mandibles on their mouth part to bite the mites. So then I kind of focus on the mandibles. The Apis mellifera are the honeybees we see here in the United States. But there's also Asian honeybees. They're called Apis cerana. And they are actually original hosts of the Varroa mites here. So I want to compare the mandibles of these bees to see if there's difference here. So we use a micro CT scanner, it's called a HaloScan, and use the right amount of laser power. You can scan the little mandibles. We put them on like a wood stick into the machine. And we get like really amazing 3D structures of the mandibles here. And for me, it's really amazing to see. Because I didn't know there's all kinds of these structures on the mandible. You can see the two mandibles, if you think about it, like a pair of scissors, right, to cut things. And so there's a long edge, short edge. There's also a bunch of hooks in here. And so we measured all these parameters, the height, the length of the mandibles. We compared the commercial honeybees with the Purdue breeding stocks. So these are high biting bees, the mite biters. And it's quite interesting on the long edge. So this is a long edge here. We see a really decrease of the long edge in the mite biters. And the commercial ones are actually longer. And so I also put I didn't put the other data, but we also have bees from AP Serrana collected in China. And we have Puerto Rican bees that are also mite resistant. So the trend is actually going down this way. So AP Serrana is somewhere lower, but the mite biters and other Apis mellifera sculata bees are actually in the middle. So the third part of my topic, I want to talk about the efforts I have put in to initiate the new land grant research at the Central State University. So Central State is a historically black university and college, and 99% of our students are African American students, underrepresented and minority. Back in 2017, when I was hired, really we started the new 1890 land grant research. So Purdue is land grant, right? That's 1862 land grant. So we're one of the few in the country for 1890. We have a lot of undergraduate student success since our B-Lab started. So we have trained 35 underrepresented minority students now since I opened the door back in the fall of 2017. So we have 8 students won the National Science Foundation scholarships, and we also have 1 student won the Control for Disease Center for Disease Control Research Fellowship. So this is an example. This is my student, current student, Jayla Marvin. She's a first-generation college student. So after she started working with our lab, and since last year, she has improved so much on her research, on her academic performance, and I just think she has a lot of more potential in the future. We also have a lot of research presentations from the students, 21 talks at ESA and branch meetings and other meetings. There are also more than 5 posters now for conference as well. So work with the students, I also try to encourage them to publish papers. So these are examples of co-authored papers with the students. Rohan is in med school now. This is Kylie, work on the oxidative stress. So I didn't have time to squeeze in the oxidative stress and physiology work in here, but I want to highlight the students here. So this is Jada, work on the biting and the mandibles, and this is Zarin. During COVID, Zarin and I worked together in one summer, and we wrote a review for the topics in oxidative stress. So I was awarded as the Faculty President's Award for Faculty Excellence in Research, and also featured on the 1 Million Women in STEM website. So the courses I've taught are entomology, IPM, honeybees, beekeeping, animal behavior, and ecology. And after I graduated from Purdue, I feel like I'm a whole Purdue entomology at Central State University. I started to look back Tom Turbin's notes, Dr. Chris Oseto's notes, everybody's notes on the classes to prepare my classes. So I give a talk in the 2021 ESA, talk about the teaching at Central State. So this is one of the highlighted comments from the attendees or member. Yeah, so I think for me, it truly, I think it's, I really want to bring entomology to Central State, and we can mentor and help to educate more people into the entomological field. So I started working actively in the Diversity Inclusion Committee with ESA. I proposed the idea to create a new award, student award for minority students. And we worked with a committee, we worked with a governing board. So it took us a year and a half, finally, to have this set up now. So I'm very happy to see a new late award. So for all the minority serving institutions, HBCUs, to support their students. It can be graduate student, can be undergraduate student. I also work with other ESA members to organize leadership happy hour and the lunch series webinar at ESA. This was during COVID time, but we were able to record all the videos and show people how to get involved with the ESA, how to volunteer, and how to be a leader, and how to get to the ballot. So as a past chair of the Diversity Inclusion Committee, I also wrote an article for the American Entomologist just to show the whole year, the last year, our achievements and activities we worked for entomology, diversity, and inclusion. So right now I'm the president-elect for the international branch ESA. If anybody have any great ideas on international branch, please let me know. We can organize awesome symposium next year for a virtual meeting. So recap, I talk about bees, I talk about DNA methylation, how to influence gene regulation. I talk about molecular mechanisms underlying the aggression and grooming behavior. So we're working on the grooming right now, so we have really cool data to show later. And then showed my efforts on new land grant research and diversity inclusion. So last, getting close to my talk, I want to call for action. A lot of you are in this room here. How many of you are ESA members? Raise your hand. Yeah, amazing. We have so many members here. Today, after this talk, go to your computer. Look for all the awards on ESA website. Faculty award, student award. Nominate amazing people you know. And nominate yourself. A lot of them are self-nominated. Let's get Purdue Entomology really amazing winners in November, right? At DC. So I really want to encourage everyone to be involved with the ESA. It's your member, it's your society. So you should involve and help. So we have fundings from NSF, USDA, a lot of amazing people help us on the research. So just bring it back to Dr. Virginia Ferris. I think her heart is still radiating in the room now. When I walk down the hallway, I think I can still feel her walking there and talk to me. I just don't feel like she left us, right? So, and I also want to share a picture of all the graduate students from 2005. Sorry, my voice is getting scratched now. So these are amazing people, grad students at Purdue Entomology. And congratulations, Hannah. I really want to congratulate you on your achievements. And it's amazing to be back and to be home as a guest lecturer. Thank you so much.
Thank you for that truly amazing and inspirational presentation, Hongmei. We are so proud of you, and I know Virginia would be incredibly proud of you, and we're so pleased to have you back here.
Thank you so much.
And thank you very much. I know she particularly would have liked the slide with the tree in it, so and all of the focus on the molecular work. But what I also know is that your contribution to training students and young scientists would really have resonated with her. So.
Yeah. So I think, yeah, with the pictures, you know, it's a legacy of her. And then, you know, back in 1950s, 60s, and then 2000s, and now we're a new generation. We're taking this legacy even longer.
Okay. I don't want to put you on the spot, but we are going to offer both of you the chance to ask the first question if you would like. But if you would prefer to pass, that's okay too.
I hope to do justice. Pass.
Okay. I have to say, the first time that Virginia didn't ask question at seminar, there was stony silence, but not like a question. We've got a couple of minutes for some questions for Hongmei. Matt.
So the mite biting behavior, you showed that there was a lot more of an likelihood of getting the mites being damaged in the hives. How does that translate into the average lifespan of a mite in a colony that can do that versus as one that hasn't been bred for this mite biting behavior?
So we actually screened the overall basic mite population between the mite biters and the commercial colonies, and there was not much difference. So I think overall the population of mites are probably maybe similar, and then if you look at the mite biters and the breeding one selection for the high grooming and the high biting, maybe because the workers in those colonies tend to respond to mites more and yeah, do more grooming.
Thank you.
One more question. Steve.
Hongmei, you've done amazing work, and your work on diversity and your work in society is laudatory. What are some things that you learned from Professor Ferris that you think you brought to your lab and the skills that you expressed today in this presentation?
I need her high energy, and you know, yeah, I mean, she's the most amazing one with the highest energy I mentioned before. It's for her, it just is like nothing can knock her down. If something knocked her down, he would get back up, you know, without delay.
Earlier than everybody else.
3 a.m., get out at 3 a.m., yeah, start working. It is I think it's the determination I learned from her. She's really determined, you know, to have graduate students do research. When we took the classes, she's very actually strict. And it was my first semester, and I have a whole thing of textbook and molecular systematics. I was like, ugh. But yeah, I passed it. I got an A, and I just worked hard with her. And every time I have a question, she would just help me, and I think I learned from her of the strong will. You're determined to do, the things you want to do, and you find a passion on the area, the topic you want to do, then you really want to put full force into it. Somebody maybe do a career, 5 years, 10 years. She did it more than 50 years. So nothing can knock her down. In those 50 plus years, she gave it all. She gave it all. Yeah. So I think that's the amazing part.
Thank you. So we're going to move to the award presentation, and so I'd actually like to ask Hannah to come up, and Hongmei to stand there, and Susan, would you like to come forward as well? Very good. This way. Thank you very much as our guest speaker, distinguished speaker, and we wanted to present you with a special award as our guest speaker. Both of the awards are flames.
Let's see it, Hongmei. All right. 1, 2, 3.
All right. Thank you, Susan. Hannah, would you like to say? Yes. So we've asked Hannah to say a couple of words. So Hannah is a graduate student working in the program of Dr. Ian Kaplan, who also happens to be the chair of our graduate committee and joined us for lunch today. And Hannah, just to talk a little bit about the impact of the award on your research.
Yeah, I just wanted to say a few words. First of all, thank you for this award. Thank you for everyone who made this endowment possible for funding my research. As we've already mentioned, Virginia and I both graduated from Wellesley College. Wellesley's a pretty small college, certainly a lot smaller than Purdue, but our alum network is very strong. And we don't produce a lot of entomologists as far as I'm aware, so it feels very special to receive our award. I really wish that I could have met her. I want to talk a little bit about my research and what I've been doing so far with the funds from the Ferris Award. So in broad scope, my research looks at long-term ecological change in Indiana honeybees, or bumblebees, and looking at changes in range and phenology, floral association, and the best resource that we have available to look at these historic changes are preserved specimens like that we have at the Perk. And we have a lot of Indiana bumblebees at the Perk, but my goal is to really enhance this collecting record for Indiana bees by going to other collections, and this involves a lot of travel. So that's primarily what I've been doing with the funds from the Ferris Award. So far, along with the collections manager of the Perk, Chris Worth, we've traveled to several different collections within Indiana and nearby states. So far we've gone to Hanover College in Southeast Indiana. We've gone down to Lexington, Kentucky to visit the University of Kentucky's collection. And recently we plan to visit out to Ann Arbor in Michigan. We're going to check out their collection and visit St. Francis along the way. We're actually very excited to go to St. Francis. That is an old personal collection of one of the nuns who used to work there, and we're pretty sure those bees have not been looked at in a very long time. So stay tuned for that. These visits so far have been very productive. We added a lot of bees to the record so far. This is just an example of one of those typical days. This is actually at Butler. They had a great collection, but typically we work very tirelessly up until the moment that we leave. But we've pulled a few hidden gems from these collections that I wanted to show off. On the right, this is a great specimen from the well-known entomologist Henry Covell from the 1940s. That was a great record to find. And that bee on the left, we just found at Butler a few weeks ago. That's a very obscure parasitic bumblebee that probably doesn't exist in Indiana anymore. So that was a delight to come across in their collection. So with the help of the funds from the Ferris Award, I found some really cool records to help enhance this data set, and then hopefully we'll be able to paint a clear picture of how our bumblebees have changed, learn more about these pollinator communities, and hopefully provide some useful information for conservation efforts across Indiana. So thank you so much.
Last but not least, we thought we would give the last word to Susan Ferris-Edgell. Thank you, Hannah, and congratulations again.
Congratulations, Hannah. It was a delight meeting Hannah today. You know, I stand here as Virginia and John Ferris's daughter. I've got an older brother, Jeff. He lives in California. He is here in spirit. He couldn't be here in person. But for us, we established this endowment to bring forward her legacy. And what about her legacy is most important? Not just the nematology, the genetics, the teaching, the research, the patents, not just that, but her mentorship of people, many of whom are in this room, I know, because I've talked to you. But we wanted to make sure that her legacy is promoted down through the next generation of emerging scientists so that the Hannahs of the world can have the boost that they need to promote their career, so that when, in a number of decades, when Hannah is a world-famous scientist, everyone knows her name in her field, Hannah will be able to push this forward and help other young rising scientists in her field. That is our wish and our hope for this endowment. So thank you all for being here today. I really appreciate it. Pete, you did a great job. Mardell, thank you for all your work. I really appreciate it. Thank you.
Well, it's been a lot of fun. Really enjoyed this, and I'm glad we were able to realize this. I'd like to close with thanking Dr. Ian Kaplan and the members of the graduate committee who were responsible for reviewing applicants for the award. I'd like to thank Dr. Matt Ginzel and the members of the research committee who were instrumental in finding a nomination and inviting Hongmei to campus as a guest speaker. I'd like to thank Dr. Hongmei Liugali as our amazing guest speaker. Thank you so much. loved seeing those pictures, and that was truly amazing. And mostly, I'd like to thank Virginia and her family for helping, for everything that Virginia has given to the Department of Entomology, the College of Agriculture, and to Purdue University, to young scientists in general, and to the field of science more broadly, and for making this this generous gift in her memory and to continue her legacy. So thank you very much. And concludes, and please